Okay, so let's start our chapter 2 discussion. For this chapter, we are going to talk about water. Water lang, okay? So, water the solvent for biochemical reactions, okay? So, what do we know? Bago tayo mag-start sa topic mismo, so this is our chapter outline. So, we'll discuss water and polarity, okay? What are the factors that can attribute to the polarity of a molecule? Then next, we will have hydrogen bonding. Basically, the intermolecular forces of attraction, if you could still remember them. And then, punta tayo sa acid, bases, and pH. And then lastly, we'll discuss buffers. Okay? Okay, so in biochemistry, water is important because andun nakadissolve lahat ng ating substances. No? Our body is composed of at least 70 to 90% water. No? Okay, pag adult person yan, that's around 60%. Pero kapag yan ay baby o kaya ano yan, halaman, prutas, no? So, mga matataas yung water contents doon. Okay? And what we know is that water is the universal solvent, no? Even in our bodies, no? So that means chemical reactions happen in aqueous medium. Okay, so ibig sabihin sa tubig siya nangyayari yung mga chemical reaction natin. Okay? Ang importance ng water sa biochemistry is that water can determine kung ano yung magiging function, ano yung magiging structure ng protein, nucleic acid, and membranes. No? Because of the intermolecular forces na ini-exhibit ng mga molecules, our protein will have its shape, no? When exposed with water, no? So magkakaroon tayo ng mga shape ng molecules dahil sa water, no? Ng biomolecules because of water. So yun yung importance ni water sa biochemistry. Number one, ito yung dissolving medium ng lahat ng substances, no? So that means all the chemical reactions will happen here. Number two, this will tell you the final shape of your protein, the behavior of nucleic acids, and different membranes of your cell. Okay? So yun, malaki yung effect ni water sa biochem. So let's talk about polarity, no? Ano ba meron na intermolecular forces? Ano ba yung mga forces of attraction na meron tayo? Review lang to muna sa ngayon. Okay, so if you could still recall our inorganic chem discussion, alam ko na-take nyo na yan before. Kung naalala nyo pa, there are two types of forces of attraction that exist, no, sa ating mga molecules. Number one is the intramolecular forces, and the other one is called the intermolecular forces. Let's differentiate the two. When we say intramolecular, intra, that means within the molecule. Okay? Kaya nga intramuros, within the wall. Intra, within, muros, wall. So dito naman, pag sinabing intramolecular, yan ay within the molecule, yung forces of attraction. So that means intramolecular forces of attraction are the forces of attraction that hold the atoms together to form a molecule. Okay? So siya yung nagsasama-sama sa mga atoms para sila ay maging molecule. So ano yung mga example nun? Covalent bond, ionic bond, metallic bond, no? So yun yung mga intramolecular forces of attraction. So ano uli ginagawa nila? They combine the atoms to create molecules. Okay, so ito yung strong type of bond. And we also call this as a chemical bond. Okay? The next type of the force of attraction is the intermolecular force of attraction. Pag sinabing inter, iba't ibang molecules. Okay, so for example, sa illustration na to, okay, so the bond between hydrogen and chlorine, so this is called the intramolecular attraction. Okay, so within the molecule, nag-exist ang bond na ito. Okay, however, it is also possible that one molecule could be attracted to the other molecule. Okay, and we call that the intermolecular forces of attraction. Okay? So ano-ano yung mga uri ng intermolecular forces of attraction? We have the dipole, we have the ion dipole, we have the hydrogen bonding, and then we have the London dispersion forces. Hopefully naalala nyo pa yun, no? Okay, so ano ba yung reason bakit may iba't ibang intramolecular or intermolecular forces of attraction? The main reason for having different types of forces of attraction is that iba-iba kasi yung polarity and electronegativity of the atoms in our molecule. When we say polarity, we are pertaining to the electrical distribution or the charge distribution in your molecule. Okay? So if your charges are distributed evenly, then ibig sabihin non-polar yung iyong molecule. Okay? However, if your charges are not distributed equally, ibig sabihin one side of the molecule have more electrons than the other, then dun nag-exist ang polarity. So nagiging polar yung molecule nyo. Later on, may picture tayo about that. So yun yung isang factor bakit iba-iba yung forces of attraction. Number one, polarity. So depende sa distribution ng electron in your molecule, the molecule could be polar or non-polar. Okay? So if ang electrons ay completely distributed, ibig sabihin equally distributed siya sa molecule, then non-polar yon. However, if ang electrons or ang charge ay isolated, ibig sabihin nasa isang side lang siya ng molecule, then dun siya nagiging polar. Okay? And as a result, dun sa separation or sa segregation ng electrons, nagkakaroon tayo ng charges, no? Nagkakaroon tayo ng poles, kaya nagiging polar ang molecule. So kung nasan yung electron, dun yung negative. Kung saan walang electron, dun yung positive, di ba? Okay? Ano pa yung factor? Bakit iba-iba yung IMFA at bakit din may polarity? Ang sagot dun ay yung electronegativity, no? So when we say electronegativity, this is a trend in which or this is a measure, no, of your atom's tendency to attract electrons towards itself, no. So ito yung tendency ng atom, hilahin yung electrons towards itself, no. Okay, so the tendency of electrons to pull the tendency of atoms to pull electrons closer to itself increases as you go up sa periodic table, papunta kang pataas and papunta sa right, no? So, dun tumataas yung tendency manghila ng electron yung atom. So, ano uli yung trend? Going up and then going right. Dun tumataas yung tendency manghila ng electron yung atom. Okay? So, the most electronegative atom will be fluorine. Noble gases do not have significant electronegativity values. Okay? So, hindi sila masyadong electronegative, no? Virtually zero yan. Bakit? Because the octet rule was fulfilled na. So, kumpleto na sila ng electron sa kanilang outer shell. So, hindi na nila kailangan manghila ng electron. And reason bakit mas mataas yung electronegativity ng mga atoms dito sa top right side ng periodic table is that they want to emulate the electronic configuration of your noble gases, no? So para magaya nila yung noble gases, kailangan malakas sila manghila ng electron. So that's why the elements here are more electronegative compared to the others here at the lower left side of the periodic table. Okay? So, yan. Periodic trends lang yan. Okay, so again, ito yung sa polarity. If our molecule is polar, that means the electron is unevenly distributed. Ibig sabihin, one side will bear the most number of electrons compared to the other side. And that can be visualized using this electrostatic potential. Kung nasan yung blue part, andoon yung wala masyadong electron. Kung saan yung red part, andun banda yung electrons. As we can see, we only have one gradient here, no? So from red to blue. So that means ito sa taas, ito yung mga maraming electron. Ano yung reason? The reason could be meron kasing electronegative atoms there. No? For example, for ammonia, which leaked yesterday sa Navotas. No? Delikado to. Sobrang baho niyan eh. So nitrogen is more electronegative than the hydrogen. No? So that's why the nitrogen will pull the electrons of the other atoms. Okay? So that's why kulay red banda kay nitrogen. Okay? Mas mataas yung negativity niya. So, siya yung hihila ng electrons, leaving virtually no electrons for the hydrogen. So, since may gradient tayo dito ng electron distribution, ibig sabihin uneven yung distribution ng electrons, polar yan. Same as with water. So, for water, yung red part ay kay oxygen because oxygen is more electronegative than hydrogen. So its tendency is that it will pull the electrons off from hydrogen towards itself. So pag hinila niyo yung electrons down, mapupunta kay oxygen yung electrons. So uneven yung distribution. So basically, that will be a polar molecule. When you say non-polar naman, so ito evenly distributed ang electron or equally shared ang electrons sa entire molecule. Okay? So for example, we have here carbon dioxide. So the Lewis structure for carbon dioxide is this one. Yeah. So the carbon is in the middle. We have 4 bonds around it. And at each end, may oxygen. Okay. So mapapansin natin, this is our electrostatic map. For carbon, there is virtually no electrons there. Kaya kulay blue. Bakit? Kasi oxygen will pull the electrons of carbon. Okay? So, hilahin niya yung electrons ni carbon on this side, on the right side, and also on the left side. No? So, if even yung distribution, meron sa left, meron sa right, ng electrons, then it is non-polar. No? So, yun. Pag meron sa left, meron sa right, meron sa taas, meron sa baba. If yung electrons nakakalat evenly sa molecule mo, then your molecule is non-polar. Okay? So, ganun. For carbon dioxide, non-polar yan. No? So, kasi hinihila ni oxygen yung electrons to the left and to the right. Okay? So, meron sa left, meron sa right, even yan. So, non-polar yan. Other forms of attractive forces ay yung ion interactions, no? So our ions can interact with other particles, no? Para magkaroon ng forces of attraction. So number 1 would be ion-ion interaction or electrostatic attractions. So basically, that's the interaction of your ion with another ion. So yan yung cation and an ion. So ano magagawa mo when you have ion-ion interaction? You have salts, no? Magkakaroon ka ng salt, no? For example, that's Na and Cl, sodium chloride, no? So, isang positive, isang negative. So, suppose we have sodium ion and then acetate ion. Pag pinagsama mo yan, sodium acetate, okay? So, ionic compounds ang mabubuo natin if we have ion-ion interactions. Okay? Pero another type of ion interaction would be the Ion-Dipole Interaction. And this is the reason why ionic compounds dissolve in water, no? At most. Okay? Not all, pero most of them natutunaw sa water. Bakit? Kasi what happens here when you place your ionic compound in water is that it dissociates into its ions, di ba? So let's suppose sodium chloride pag nilagay mo yan sa tubig, sodium chloride will dissociate into sodium cation and chloride anion. Then what will happen is that since water is polar, okay, so polar yung ating water because it is dipole. Ano uli meaning ng dipole? Dalawa yung poles, may positive, may negative, no? So yung negative pole, dun yung electronegative atom. Sa positive pole or the positive end, dun yung least electronegative atoms. Diba? So water is dipole and it is polar. No? Kasi yung oxygen, andun yung partial negative charge kasi siya yung pinaka-electronegative na atom sa molecule. Hydrogen will bear the partial positive charge kasi siya yung least electronegative atom. Okay. So kung mapapansin mo, yung water natin, may partial charges siya. And those partial charges can actually be attracted to your ions. No? So, kunwari, yung salt, nilagay mo sa tubig. What happens there is that yung salt, nagdi-dissociate siya into ions and then yung water molecules, papalibutan niya yung ions natin. Okay? So, let's suppose we have sodium cation here. Yan ay papalibutan ng oxygen and ng water molecules. No? Bakit oxygen and? Kasi oxygen is partial negative. Sodium is positively charged, no? So may negative, may positive, there is an attraction. We call that ion-dipole interactions. So for chloride naman, ano yung attracted sa chloride ion? Ang partial positive end ng water molecule. So may partial positive, may negative, positive negative attracts. So ano tawag doon? Ion-dipole interaction. Okay? Excuse me. So that's the reason why salts, no, ionic compounds dissolve in water because of the presence of dipole in water, especially in water molecule mismo, dipole yan, and yung ions, yan. So may interaction yung kanilang charges, no. So pwede sila mag-dissolve. No? Pwede niya i-dissolve yung salts natin. Okay. And then next one, we have the intermolecular forces naman. So there are three types of intermolecular forces in general. So that's van der Waals forces or London dispersion forces, the dipole-dipole interactions or forces, and the hydrogen bonding. No? Of the three intermolecular forces, hydrogen bonding is the strongest. No? Okay, so that's experimentally determined, no? Na mas mataas ang strength ng hydrogen bonding compared to other intermolecular forces. Okay, so let's talk about the three. No? When we say London dispersion forces, these are forces of attraction that doesn't depend on the polarity of your molecule. As long as it has electrons, it can exhibit London dispersion forces or van der Waals dispersion forces. No? If you could still remember yung kwento natin nung org-chem about the instantaneous dipole, yun sa London dispersion forces or sa van der Waals forces. Yung ating electrons sa ating molecule, it could travel, no? And kapag nagpungguan yung dalawang molecule, pwedeng magkaroon tayo ng temporary dipoles, no? So, kunwari, we have this, oil molecule, for example, and this one is another oil molecule. Pag pinagdikit mo yan, pwede itulak nito yung electrons ng kabilang molecule papunta sa other side. So that will have its partial negative and partial positive end. No? Doon sila pwede maging attracted. However, that is just temporary. Temporary lang kapag van der Waals forces, no, or London dispersion forces. Temporary lang yan. Mawawala din yan. That's why it is the weakest intermolecular force. So ano uli meron kapag London dispersion forces or van der Waals forces? Ang pinag-uusapan lang ay electron. And what happens to those electrons is that they create instantaneous dipoles. Tama ba spelling ko? Okay, so they create instantaneous dipoles. Ibig sabihin, they create temporary dipoles. Pag nagdikit yung dalawang non-polar molecule, yan, pwede magkaroon sila ng temporary dipole. Okay, so pag nagkaroon sila ng temporary dipole, they could be attracted temporarily. However, it should end then. So pag nag-iwala yung molecule mo, back to non-polar sila. Okay? So that's the London dispersion forces or the van der Waals forces of attraction. Next one is the dipole-dipole. So this is the attraction between dipole molecule and another dipole molecule. So remember, when we have dipole, we have partial positive and we have partial negative ends for our molecule. Those partial charge partially charged ends ng ating molecule, they could be attracted to one another, just as shown here. So for example, we have this hydrogen sulfide. So yung negative end ng molecule na yan, that could be attracted to the partial positive end of the same molecule, no? So pwede silang magdugtungan sa dulo-dulo ng molecule. And we call that dipole-dipole interaction. However, if your dipole contains hydrogen bonds, so ano uli yung mga hydrogen bonds natin? Yung OH, NH, and then F FH bonds. If your molecule consists at least one of them, then it can qualify dito sa hydrogen bonding. So, yun. It is experimentally said na mas mataas or mas malakas ang hydrogen bonding compared to dipole-dipole. No? Ayan. Because this type of intermolecular forces ay special kasi sa kanilang tatlo lang to sa kanilang tatlo lang yan observed, no. So yun. So one example would be here. We have water and ammonia. So this is water molecule. So you can see meron yang OH. This is ammonia. We have NH, no? So that means the OH and the NH end of your molecule can actually be attracted to one another. So same concept lang siya as dipole-dipole. Ang exception lang is meron kang dapat OH, NH, FH bond for you to have hydrogen bonding. Okay? So ganun lang. So yan yung review ng ating inorg chem. So nag-review lang tayo sa ngayon, no? So basically, inalam muna natin paano ba natin nasasabi na polar ang molecules, no? So the factors that can affect the polarity of your molecule is the electronegativity and the distribution of electrons in your molecule, no? And then these are the types of the interactions that was discussed before during your inorganic chem. So, nireview lang uli natin. No? Dito natin nakita that salts can actually dissolve in water. No? That's why our body contains salts then. May mga salt tayo sa katawan. No? Such as sodium, potassium, chloride. No? So, may mga ganyan tayo sa ating katawan. Okay, and then ito naman, kwento natin yung intermolecular forces of attraction. So, ano yung tatlong uri nila and what are the differences? Okay, so mamaya kwento natin yung application niya sa biochem. So, daan muna tayo. Okay, so si water is a polar molecule. Okay, so looking at the structure of the water, we have 4 electron pairs, which is on a distorted tetrahedron geometry. So, asan yung 4 electron pairs? 2 of which ay naka-lone pair. And then the other 2 electron pairs ay in the form of chemical bond, no? The OH bond. Okay? So, kung kukunin mo yung kanyang molecular geometry, that is actually a distorted tetrahedron. Pag kinuha mo yung electron, geometry, that's bent. Okay? So, our central atom here would be the oxygen, and then our terminal atoms would be hydrogen. Okay? And since oxygen is more electronegative than hydrogen, then we say that it will pull oxygen will pull the electrons of hydrogen, creating it the partial negative end of your molecule. So, yung hydrogen, that will be the partial positive end of the molecule. And because we have uneven distribution of electron, na kay oxygen lang yung electron, that means we have a dipole moment. No? So, dipole moment is the direction of your electrons. No? So, we can see here that the electrons are traveling towards oxygen. So that means the dipole moment is towards oxygen kasi siya yung may electrons. Okay? So, ayan. Ano yung importance nito? Ano yung importance ng structure ng water sa biochemistry? So, una, so this is the hydrogen bonding water. Water can serve as both hydrogen bond donor and acceptor. So that means because of the presence of the OH bond, it can form hydrogen bonds with other molecules such as biomolecules and water. Okay? And because of that interaction of water with other H bond forming molecules, marami siyang effect, no? Ito yung importance ni H bond ng water. Okay? So, maraming effect yun sa ating mga biomolecules. For example, hydrogen bond is important because it shows the unique properties of water. That is a small molecule pero malakas yung uh, strength ng mga molecules na yun. That's why it boils at 100 degrees Celsius compared to others. And also, hydrogen bonds can determine the structure and the function of your proteins. No? So kapag yung protein na buo, pumunta yan ng water, let's say sa cytoplasm, pag nasa cytoplasm siya, it will fold into its final shape. Then, yung final shape niya, that will determine its function. For example, yung ating red blood cell, that is a protein. No? That's hemoglobin. So, yung protein na yon, it is shaped like a disc because of the effect of the hydrogen bond. No? So, ganun. Other functions and structures affected by H bond would be the DNA. So imagine nyo, paano naging double-stranded ang DNA? Because at each strand of your DNA, it contains the nucleotides, no? Okay, so may mga nucleotide bases tayo doon. Okay, so what happens is that nagdudugtong sila, no, to form hydrogen bond, no. So ito yung example nun. So this is one strand, ito yung isa pang strand, and they are connected in the middle of the sa middle ng twist, no, sa helix. They are connected sa middle with hydrogen bonds, no. Okay, so paano yon? So 'di ba alam natin cytosine and guanine sila magka-partner because equal amounts ng hydrogen bond yung kaya nila gawin. Okay? So, oxygen can bond to NH, N can bond to HN, N can bond to O. Okay? So, yun yung mga pwedeng site for hydrogen bonding. That's why cytosine and guanine are tandems. No? Magka-pair sila parate. Other pair is the thymine and adenine. If you are talking about RNA, this will be uracil. Okay? So, ganun lang. Pwede yung H bonding dito, NH and N, and then O with NH. Okay, so because of the presence of NH and OH bonds, then it can form hydrogen bond. So that is the reason why our DNA is stranded. No? H bond yung dahilan bakit siya stranded. Okay, and then next one, hydrogen bond can determine the structure and the function of polysaccharides. When you say polysaccharides, that's basically sugars, no? So sugar can come into many form. It could be monosaccharide, disaccharide, oligosaccharide, polysaccharides, no? So an example niyan, yung let's say yung mga dagta, no? Pectins, 'yan. Bakit siya madagta, no? Although madi-discuss din natin 'yun in the future chapter, yung reason ba't siya malagkit is hydrogen bond, no? Kung naalala nyo pa yung in-org discussion nyo last time, hindi kasi ako yung in-org nyo, di ba? Kung naalala nyo pa yung in-org discussion about viscosity, yan. So, hydrogen bonding, yung malaki yung effect doon. So, for example, honey, no? Yung honey, compare mo yan sa water, sino mas malapot? Yung honey, because it contains more hydrogen bond than water. Okay? So ganun din sa mga saccharides natin, no? Let's say mga dagta, no? So yun, malagkit sila kasi may hydrogen bonds na marami doon. Okay? Pero kapag nababawasan yung hydrogen bonds, no, let's say asukal lang, okay lang yung lagkit niya. Okay? Next one, another importance of H bond sa biochemistry would be the binding of the substrate to enzymes, no? Paano yan nangyayari? Kunwari, you have your molecules, no? Didikit siya sa enzyme for it to be catabolized. Ibig sabihin, magamit in biochemical reactions, no? So, pag yung molecule na yon didikit siya sa enzyme, it sometimes form hydrogen bonds in the active site of that enzyme, no? More of that will be discussed sa chapter 5 naman. So, leave that hanging na lang muna. Okay? So, yung substrate natin, ito yung mga molecule na kumakabit sa enzyme. Paano siya kumakabit doon? Through hydrogen bonds na. So, kakabit siya by hydrogen bond, then saka siya gagamitin ng enzyme to form the products. Okay? And another importance would be binding of hormones to receptors. This is important for biosignaling, no? Kunwari, marami ka ng sugar. So, your cells can actually trigger the the production of insulin, no, para ma-lower yung blood sugar levels mo, no? Ma-control yung blood sugar levels, no? Or kunwari mababa yung sugar mo, then the cells, no, could trigger the gluconeogenesis pathway. So, ang mangyayari doon, yung fats stored sa katawan natin, they will be converted to sugar. Okay? So, yung process ng signaling ng ating mga hormones with the receptors in our body, they are governed by H bonding. So, yung pag-signal ng mga molecules sa katawan natin, sa tulong ng hydrogen bonding nangyayari yan. Okay? And then lastly, we have the matching of mRNA and tRNA that is important kapag tayo ay gumagawa ng proteins. No? So, yan. So, yun. So, sila yung mga ginagamit to create proteins, no? And other molecules sa ating katawan. Okay? So, kung mapapansin natin, ang daming ganap ni water sa ating katawan na. Okay? So, yan. What else do we have to know about water? Okay, so water is a good solvent for charged and polar substances such as amino acids, peptides, small alcohols, and carbohydrates. No? So, ayan yung itong mga to, these are all present in our cells. No? So that's why our cell contain water. No? So they dissolve all these substances, no? para mag-function yung cell. However, water is a poor solvent for non-polar substances kasi these are non-polar molecules, so hindi sila magmi-mix, no? So, hindi sila maghahalo. Kaya poor solvent siya for non-polar solvents such as non-polar gases, aromatic moieties, no? When you say moieties, yung mga aromatic structure, yung mga may benzene ring. Okay? And then aliphatic chain or mga hydrocarbons. So these are all non-polar molecules, so they don't dissolve very well in water. However, these molecules are polar and some of them can form hydrogen bonds such as alcohol and carbohydrates. So they are dissolved very well in water. Okay. So itong mga to hindi kasi they do not possess dipoles, no. Hindi dipole tong mga to. So they will not dissolve in water very well. Okay? And ito pa yung isang important na effect. Ito pa yung isang important na ganap ni water sa biochem, the hydrophobic effect, no. So ang nangyayari kasi if our amino acids form long chains, no, magiging peptide yon, and then later on kapag dumami na yung chain ng ating amino acids, siya ay nagiging proteins na. Okay. So yung proteins, since malaking molecule na yon, it becomes non-polar at times, no. Okay. So what happens is that kapag yung iyong protein, so let's say ito yung ating protein, if yan ay nilagay mo sa water, what will happen is that it will contort its shape. Ibig sabihin, magbabago yung shape niya pag nilagay mo yun sa water in such way that the hydrophilic ends are pointed away from the molecule. No? So, ito yung molecule natin. Ito yung ating ito yung ating protein. Mayroon yung hydrophobic and hydrophilic ends. Yung mga hydrophobic ends, yun yung mga aliphatic chains sa ating proteins. Yung mga hydrophilic, sila yung mga may hydrogen bonds or mga dipole. So, nangyari kapag nilagay mo yung ating protein na to sa water, mag-contort yan to form certain shapes na in such way that the hydrophilic end is away and yung mga hydrophobic end, sila yung magka-clump inside, no? So, that's why our protein has their own shapes, no? Such as enzymes, may sarili silang shapes, no? Because of the hydrophobic effect. Ganun din sa sa ating, let's say, red blood cells, no? So, yung shape niya nagbabago-bago depending on the water that is around it, no? Let's say kapag sobrang daming tubig sa paligid, nagbabago yung shape ng ating red blood cell, lumulobo siya. Kapag masyadong onti lang yung water sa paligid, nag-shrink siya. So those shapes of our proteins, yun ay dahil sa water, especially the hydrophobic effect. No? So ganun lang. So yun lang yung importance ni water sa biochem. So again, ano yung importance ni water sa biochem? Um, Water can serve as H-bond donor and H-bond acceptor. So that means pwede siyang makaapekto sa iba't ibang molecules sa biochemistry na may H-bonds. No? So H-bond can determine the function of water, the structure and the function of proteins, yung sa DNA, polysaccharides, no? How substrates attach to the enzymes when it comes to enzyme catalysis. No? And then kung paano nasi-signal yung ating paano nasi-signal ng katawan natin yung mga cells to do stuff, no? And lastly, yung protein making. So, H-bond may kasalanan sa lahat ng mga bagay na iyan. Okay? And then, yun, water, pwede siya mag-dissolve ng polar molecules. However, if yan ay non-polar, hindi yan madi-dissolve na. And yung effect ng water when you add proteins into it is that it will allow for the protein to contort to its final shape na. So mag-arrange siya ng sarili niya to form its final shape na. So yun. Yung mga binabado kong stuff, madi-discuss pa naman natin in the future chapters. Yung mga shape ng protein, madi-discuss ko pa yan, no? Chapter 4 yan. Yung enzymes, madi-discuss ko yan. Chapter 5 yan. Carbohydrates, madi-discuss natin yan. Chapter 9 yon. So, don't worry. No? Ang gusto ko lang muna makuha nyo is yung mga pointers lang na malaki ang epekto ng H-bonding and ng water sa biochemistry. So, we will discuss them in depth when we are in those chapters na. Okay? So, for now, let it be an idea lang muna. Okay? So, ganun lang. Okay? So, next one, we'll proceed with the acids and bases and the pH. No? So, for acids and bases, there are lots of definitions. However, yung basic lang yung gagamitin natin for biochemistry. Okay? So there are three major definitions for acids and bases. The earlier definition would be from Arrhenius. Okay, so according to Arrhenius, acid produce H plus ions and bases produce OH minus ions. So again, acids produce hydronium ions or H plus. Pag bases naman, they produce hydroxide anion. Okay, so yun. Yun yung pinaka-basic. However, nag-improve yan with the Bronsted-Lowry definition. Ito yung gagamitin natin sa biochemistry. So, according to Bronsted-Lowry definition, acids are those who donate protons. When you say proton, hydrogen lang yan. So, when your molecule donates hydrogen to the other molecule, then that means the original molecule is an acid. No? So, for example, we have this HA. Pag nakita nyo sa reaction, nawalan siya ng H, then that means it is an acid. Okay? Kung sino yung mawawala ng H, siya yung acid. And then the bases, those are the proton acceptor. So kumbaga, if you will check the equation, kung sino yung madadagdagan ng H sa dulo, siya yung base. Okay? And then, once the acid donate its hydrogen to the base, it becomes the conjugate base. No? Okay. So, yun yung tawag. Kapag yung acid nawalan ng H, binigay niya sa base, ano na tawag sa kanya? Hindi na siya acid. We call that the conjugate base. We call that the conjugate base na lang. And then when our base accepts the hydrogen from the acid, it is now called the conjugate acid. Okay? So, yan yung definition na gagamitin natin sa biochemistry. Other definition is the Lewis definition. Okay, so according to Lewis, acids are those who accept electron pair and bases are those who donate electron pairs. Sa inorganic chem na yan madalas. Okay? So for now, ito yung gagamitin natin. Again, so how will you classify whether a molecule is an acid or base? Just look at the equation. Kung sino yung nawalan ng H, siya yung acid. Kung sino yung nakatanggap ng H, siya yung base. And again, we call the molecule that has its hydrogen removed the conjugate base. Yung natanggalan ng hydrogen, conjugate base tawag doon. And yung nagdagdagan ng hydrogen, we call that the conjugate acid. The conjugate acid. We can classify the strength of acids depending on its acid dissociation constant. Okay, so the acid dissociation constant tells us about the degree of dissociation of the H with its anion. Okay, so if our acid dissociation constant is high, then that means our acid is strong. So, malakas yung acid kapag na papamigay niya agad yung H niya, yung hydrogen. Pero if hindi ganun niya napapamigay, ibig sabihin, weak acid siya. Excuse me. So ito yung example natin. We have these acids here and these are their Kas. So compare lang natin. Kapag ang acid mo ay strong, that means your Ka is very high. So in this case, that's 2 times 10 raised to 5, which is 20,000 ata, no? So 20,000 yung Ka niya. Compare that with water. The acid dissociation constant for water is 2 times 10 raised to negative 16 for this example. Okay? So, ito naman, this is acetic acid. Ito yung nasa suka. So, that is a weak acid. No? The Ka is 1.8 times 10 raised to negative 5. Okay. So, yun, medyo mababa pa yan kasi decimal yan. So, that means anything na decimal yung Ka natin. So, that means negative yung kanyang scientific notation dito. Negative 4, negative 5, and so on and so forth. Basta negative yon, then that means our acid is weak. However, if our Ka is greater than 1, okay, for example, 25 or 20,000, then that means your acid is very strong. Okay? How do we solve for the Ka ba? Hindi ko alam kung may analytical chemistry kayo, pero ganito mag-solve ng Ka. Hindi naman natin gagawin to, don't worry. So how do we determine the experimental value for Ka? Basically, if we have an equilibrium equation, equilibrium equation? That is an equation with two arrows. That means two processes ang nangyayari. Reactant converting into products. Products being converted back to reactants. So if you have an equilibrium equation, then to solve for Ka, you just determine the concentration of your products over the reactants. Pag nakuha mo yung kanilang concentrations and then you divide them, you will get the Ka. So, ganun po namin siya kino-compute. So, that's why we were able to come up with these values here. So, yun lang. Again, kapag mataas ang acid dissociation constant or Ka, that means your acid is very strong. Kapag mababa ang iyong acid dissociation constant, that means decimal yan or negative yung exponent sa scientific notation, then your acid is weak. Okay? So, weak acid lang siya pag ganun. How do we solve for Ka? Ano lang yan? Products over reactants lang. Okay? Mamaya, madadaanan pa natin to. Anyway, so we're done with acids and bases. Ngayon, let's talk about pH. Okay. Medyo maingay lang dito. So let's talk about the pH. When you say pH or power of hydrogen, that is just the negative logarithm of the hydronium ion concentration. So in equation, pH is just the negative log. That is negative log of the concentration of your acid. Okay. And then with this equation, we were able to come up with the pH scale, no? And usually, alam natin, yung pH scale ay nagre-range from 0 to 14, okay? Although it can exceed that value, no? Pwede siyang maging negative or pwede siyang greater than 14 naman, okay? So, ito yung ating pH scale for example lang. So, what we know about the pH scale is that if your pH is 7, that means your molecule is neutral. Okay? So, kapag ang pH mo is 7, yan ay neutral. Kapag ang pH mo ay below 7, that means acidic yan. Kapag ang pH mo ay greater than 7, then that means yan ay basic or may base dyan. Okay? So, ganun lang. The counterpart of pH is the POH, no, or the power of hydroxide. And whatever the value of pH and POH is, kapag pinag-add mo sila, dapat equal sila sa 14. Okay? So, they are always equal to 14 pag pinag-add mo yung pH or yung POH at 25 degrees Celsius. Okay? So, ganun lang. For example, kapag ang pH mo 7, your POH is 7 din. So, pag pinagad mo yan, that will be 14. For example, your pH is 4. That means your POH is 10. Pag pinagad mo yan, 14 din yan. Okay. So, yun lang. So, yun. Again, our pH is just the negative log of the concentration of the hydronium ion. Okay. And that scale ranges from 0 to 14, although it can exceed sa range na yan. So, pwedeng maging negative, pwedeng maging greater than 14. Again, yung counterpart ng pH ay yung POH, no? And when you add pH and POH, it should always be equal to 14 at 25 degrees Celsius. Okay? So, ayan. So let's try some examples here. Problem. Number 1, what is the pH of stomach acid? A solution of hydrochloric acid with hydronium ion concentration of 1.2 times 10 raised to negative 3 m. Ano yung m? I don't know if na-discuss to sa inyo. This is molarity. So molarity is a concentration unit, no? Okay, so concentration unit yan. It tells you how many moles of acid are there in a liter of solution. So that's molarity. That is mole per liter. So kung hindi pa kayo aware sa concentration units, pwede punta kayo sa YouTube playlist natin, AY221 Chap S2 plus recordings. Check nyo yung MTY1103 Chapter 1 Laboratory. So, dun discuss yung solutions eh. Kung gusto nyo lang naman. Okay? Pero anyway, so dito yung concentration ng acid na sinasabi natin sa pH. So, when you say hydronium ion concentration, the unit must be molar. Molar dapat or mole per liter. So yan dapat yung unit kapag kukunin mo yung hydronium ion concentration. So according to problem number 1, so since the pH is, the hydronium ion concentration, so that is written as the square bracket of H plus, yan yung concentration of hydronium ion. Since that is equal to 1.2 times 10 raised to negative 3 molar, ano daw yung pH niyan? So to solve for its pH, ganito lang gagawin nyo. pH is equals to negative log of the H plus concentration. Gagawin mo lang is whatever the number for your molarity is, kung ano yung value niya, yun lang ilalagay mo sa loob ng logarithm function. So that's pH equals negative log ng 1.2 times 10 raised to negative 3. Pakicalculate na lang. Okay, so the pH is 2 decimal places tayo. That will be 2.92. So yan yung value ng pH natin for the stomach acid na. Yan yung nasa loob ng sikmura natin. Kaya 'di ba kapag kayo nagkakaroon ng acid reflux, or bigla na lang kayo nasusuka, maasim, 'di ba? Kasi nga acidic yung nasa sikmura natin. Okay. So ganun. Follow up ko lang dito sa number 1. What will be the POH? Ano kaya si PO o si POH? Again, recall that pH and POH is always equals to 14 at 25 degrees Celsius. So if you want to solve for POH, that is just 14 minus the value of pH. Okay? So that will be 14 minus 2.92. Your POH will be 11.08. So pag excuse me, pag pinag-add mo yung POH and yung pH, that will always give you 14. Yun lang.
Sir, may unit po yan.
Walang unit yung pH. Value lang siya. Number lang. Kaya tinatanggal natin yung concentration unit dito. Number lang kasi siya. Nakuha nyo naman sa calcu. Okay. So, yan. So, hopefully, nasulat nyo na. Let's have next one, number 2. Calculate the hydronium ion concentration of blood, the pH of which is 7.3. Paano yan? Ang alam natin, from concentration to pH, we can use this equation. Diba? So, from concentration, pwede natin siyang gawing pH. Ang question number 2, from pH, gagawin mong concentration. Paano yun? Okay, so ganito lang. So, according to logarithm function, gawin ko yung derivation. So, if you want to get the concentration of your H plus, of your acid from the pH, you have to rewrite the equation. Okay? So, kailangan tanggalin natin yung log. Kailangan mawala yung log. Okay? So, pakita ko muna yung derivation. Pero yung final equation, yung kailangan lang natin. Okay? So, ang una mo daw gagawin using this equation, you multiply them both sides by -1 so that yung negative mapupunta na dito sa pH. So, yung log magiging positive na. Okay? So, what's next? Okay, para ma-cancel mo yung log, kailangan gawin mo siyang exponent. Okay. So, that means you have to make this number an exponent. Ano yung base mo? ano yung number sa baba ng exponent. So, that will be 10. Okay? So, ano yung gagawin mo dito? So, yung log, gagawin mo siyang exponent ng 10. So, kung anong ginawa mo sa right side, yun din gagawin mo sa left side. Okay? So, para equal pa rin yung ating equation. Okay, bakit? Kasi yung log daw and yung 10, no, kapag ginawa mo sila ng exponent of each other, they tend to cancel out. Okay? So, that's according to algebra. So, yung 10 pati log, magka-cancel out sila. So, magka-cancel out and that will leave you the concentration of H plus na lang. So, ito yung final equation that we need, no, to solve for the concentration from the pH. This is the anti-logarithm. So, yun yung ginawa natin dito, anti-logarithm. Okay? So, yung base ng logarithm natin, yun yung ginawang base ng exponent. So, ganun daw sabi ng algebra. Okay? So, ito yung equation na gagamitin natin for number 2. If you want to convert pH to concentration. Okay? Siguro with that equation, kayo na lang magsagot ng 2 and 3. Okay? Ayan, so sagot na lang kayo 2 and 3. I'll be back mga 5 minutes lang. Okay, so wait ko yung answers nyo sa chat box natin. By the way, may nakalimutan ako sabihin pala, no? For number 3, you're asked, what is the pOH, no? Ang given sa inyo sa number 3, burahin ko muna tong sulat ko dito. So, ang equation natin, negative log, concentration ng H+. Then, from concentration ng H+, so ito na yung equation natin. If you want to get the pOH, no? Kunwari, given sa iyo yung concentration ng hydroxide, which is in our last example, yun yung case. Okay, so ang given sa inyo is concentration ng potassium hydroxide. Hydroxide, ibig sabihin base. So if you want to solve for the concentration of base to pH, ano gagawin? I mean pOH mo, gagawin mo. POH is equal to negative log ng OH. Okay. So ano yan? Yung H and yung OH, pwede mo sila pagpalit-palitin. Okay? So ito pH log H, OH log OH. Okay? So ganun din dito. If you want to get the concentration of OH, then that is just 10 raised to negative POH. So interchangeable yung ating terms. Okay? So yun yung magagamit nyo for number 3. Okay? Sagot uli kayo.
Pero sir, pwede rin ba sir gamitin yung unang formula tapos ima-minus mo na lang yung 14 dun sa pH?
Pwede naman. Pero kung ito yung gagamitin mong equation, H plus kasi ito eh. Ang meron tayo dito ay hydroxide. So,
Okay.
kailangan OH talaga dyan.
Okay.
So, ito yung gagamitin mo. Okay? Type nyo na lang yung answer. Sagutan natin. Okay, so 38 na. So, sagutan na natin. Okay, so let's have number 2. Let us calculate for the hydronium ion concentration of blood whose pH is 7.3. So for number 2, the equation to be used will be concentration of H plus equal to 10 raised to negative pH. Okay, so that's H plus equals 10 raised to negative 7.8. Ay, 7.3 pala. Teka lang, erase ko. So that's 10 raised to negative 7.3. So the answer will be 5.0. Since maraming numbers, kahit yung 2 decimal places lang. So 5.01 times 10 raised to negative 8. So that is the concentration of the hydronium ion sa ating blood whose pH is 7.3. Ano yung unit na? Kapag concentration, kailangan may unit. Kapag pH, POH, walang unit. So ano yung ilalagay nating unit dito? So katulad ng sa previous problem, pag concentration, ang ginagamit nating unit ay molar. Okay? So you can represent that by capital letter M. Okay? So pwede natin isulat yan as capital letter M. Pwede rin naman na mole per liter. So we can write that as this. Okay? So yan. So again, kapag pH, POH, walang unit yan. Pero kapag concentration, kailangan may capital M, which means molarity. Pwede rin naman mole per liter. Okay? So ganun lang. For number 3, we have here 0.0125 molar solution of potassium hydroxide. So, ano yung concentration ng hydroxide dito? Kasi ang ating salt ay potassium hydroxide. So, that means hydroxide yung meron tayo dito, not hydronium ion. Okay? So, ano yung concentration ng hydroxide dito? E di kung ano yung nakalagay doon sa ating problem. So, that will be 0.0125 molar. Okay? So that is the concentration of OH. So if you want to solve for the POH, ano gagawin natin? So POH is equals to the negative log of the concentration of OH minus. So that will be negative log of 0.0125, and that will give us 1.90. Yan yung POH. Okay. So, bakit POH kinuha natin? Kasi ang meron tayo dito ay concentration ng OH. Ito yung concentration ng OH. Paano mo malalaman? Kapag yung iyong salt ay hydroxide. Basta may hydroxide, OH yan. Okay. So, pag may makita kayong hydroxide, OH agad yan. So, ganun. Kapag gusto mo naman kunin yung pH for this problem, that will be just 14 minus POH. Okay? Kasi POH plus pH is 14. Rewriting that equation, you will get pH equals 14 minus POH, diba? So that will just be 14 minus 1.90. That will give you 14. That will give you 12.1. Okay? So, basta makita nyo hydroxide, OH agad yan. So, masasolve mo doon, POH. Okay? Then, kung gusto mo kunin yung pH, doon ka lang mag 14 minus POH. Pero kapag sinabi dyan, hydronium ion, pH, makukuha mo dyan. So, ulit-ulit lang naman yan. Okay? Ayun, screenshot nyo na lang yung answers natin. Okay? So, let's proceed. Buffers, last na tayo. Okay. So, what are buffers, no? So, 'di ba naririnig nyo, buffer zone. Kumbaga, parang this is a stabilized zone, no? Okay. Pag buffer zone, buffer quiz, no? Pampa-balance. So, sa chemistry, sa biochemistry, may ganun din, no? So, when we say buffer solution, it technically means that it is a solution that contains weak acid or weak base and its conjugate acid or base or its salt, no? So ano ba yung importance nitong buffer? So buffer helps us to maintain the pH, okay? So nakakatulong siya para hindi magbago yung pH agad-agad. And buffers are present sa ating katawan, okay? Bakit? Kasi imagine nyo, if walang buffer yung katawan natin, uminom ka lang ng suka, super acidic ka na agad. Which is uminom tuloy ng suka. Wala, magsawsaw ka lang ng suka. Dapat super acidic ka agad, no? Pero hindi ganun yung case, no? Our body tries to balance the pH pa rin, no? So, ganun. So, ibig sabihin ang buffer, these are solutions that maintain the pH, no? So, mini-maintain niya yung pH value. Okay. So, tinatry niya maging constant yung pH nang hindi nagbabago bigla-bigla. Because without buffers, malaki yung changes agad sa concentration or sa pH natin. Okay? So, yan. So, ano yung composition ng ating buffer solution? So, our buffer solution must contain the acid or yung base and its conjugate acid or base. Okay? So, kailangan may acid ka and the conjugate base. Or kung may base ka, kailangan may conjugate acid ka. Okay? Paano mo uli malalaman yung relationship ng acid sa conjugate base? Yung conjugate base, that is just a molecule with one less hydrogen. Okay? Kunwari yung acid mo, CH3COOH. Okay? Kunwari yun yung acid mo. So, ano yung conjugate base mo? So, that will be CH3COO. Yun lang. Yeah, so, isang H lang yung difference nila. Okay? So, kailangan magkasama sila sa solution for you to be able to make buffer solutions. Those are solutions that resist the change, the gradual change sa pH. Okay? And buffer solutions are governed by the Henderson-Hasselbach equation. So itong equation na to, ito yung pinaka basis, no, in determining kung ano yung capacity ng buffer natin. Meaning ng capacity ng buffer kung saan siya mas stable. Meaning nun kung saan mas fix yung pH niya. Okay? So this equation will tell us kung ano yung magiging resulting pH if we mix different concentrations of our acid and the conjugate base. Kapag pinaghalo mo yung dalawa, you will create a buffer solution. Okay? So, ganun. So, let's try this problem. By the way, ang nakalagay dito, it could be concentration. Pwede rin siyang moles. Mabilisan lang naman tayo. Hindi naman tayo magfo-focus masyado dito. Siguro ngayon nyo na lang ulit ito makikita, yung buffers. Next chapter, hindi na. More on kwento na uli si Biochem. Okay, so again, Henderson-Hasselbach equation, they determine what pH your buffer solution is. Na. Ano yung pH ng buffer solution mo? So siya yung magsasabi. Si Henderson-Hasselbach equation. Okay. So, let's try this problem. Dalawang problem lang meron tayo dito. Kasi hindi naman natin ito i-focus masyado. Dadaanan lang talaga natin to. Okay, so you need to produce a buffer solution that has a pH of 5.27. You already have a solution that contains 10 millimoles of acetic acid. Excuse me. How many millimoles of sodium acetate will you need to add to this solution? The pKa of acetic acid is 4.75, 4.752. Okay? So basically, according to the problem, I want to create this buffer solution. Yung pH daw ng buffer solution ko is 5.27. Kunwari, yun yung gusto ko gawin. Kunwari, I am working with enzymes, no? Tapos yung enzyme na yun, it performs best at this pH. So that means, kailangan ang pH ng solution ko ay 5.27. Kasi any pH other than that, hindi gagana yung aking enzyme. So I have to maintain this pH. So if I want to create this concentration of of my buffer, and I already have 10 millimoles of my acetic acid, ilang millimoles ng sodium acetate ang kailangan ko idagdag para ma-maintain tong pH na to, given that the pKa is 4.752. Okay, para mag-make sense yung numbers, isulat natin yung equation. Okay, so yan. So this is the Henderson-Hasselbach equation. That is pH equals pKa plus log ng concentration ng conjugate base natin. Okay? Ano yung acid natin? That will be your acetic acid. And this is the acid. Okay? So again, remind ko lang, yung ating conjugate acid, conjugate base, pwede silang in terms of concentration. Pwede ring moles. In our case, millimoles. So, okay lang yan. So, yan. So, we already have the acid. We have 10 millimoles of acetic acid. Ano yung conjugate base ng acetic acid? Edi yung acetate. Okay? So, ganun yung usual na nangyayari. Sodium acetate, acetic acid. So, yun yung mga partner. Mas madali yan makita kapag may chemical equation ka. No? So, ang acetic acid that is written as CH3COOH, and sodium acetate that is written as CH3COO-Na. Yung Na, ion yan, so mawawala yan. You will be left with CH3COO minus lang. So, still the conjugate base. So, yung gagawin nyo kapag kunwari given is yung formula, yung cation ninyo, tanggalin nyo lang yan. Yung metal, tanggalin nyo. Whatever is left, yun yung inyong conjugate. Conjugate base or conjugate acid. So, yan. Ngayon, plug in natin yung values. Ang sabi natin, kailangan ito yung resulting pH. So, kailangan ang result ng Henderson-Hasselbach equation natin will be 5.270. Ang pKa, that is always given. So, that will be 4.752. Log, that is a function. We already know the concentration of the acid, the moles of acid, which is 10 millimoles. Ang question na lang dito is, ilang millimoles yung conjugate base mo. Okay? So, ano daw kaya yung concentration ng conjugate base dapat? So, kailangan gamitin natin dito yung mga ginawa natin sa logarithm kanina. Okay? So, let's solve for that. Clear ko lang ulit yung screen. Zoom tayo dito sa clear part. So, we start with the Henderson-Hasselbach equation. Okay. So, ang hinahanap natin ay yung conjugate base, no? Yung millimoles nito. So, ito yung hinahanap. So, that means we should write an equation that is exclusive for conjugate base. No? So, that means anything else na kasama niya sa side ng equation na to should be written at the other side. Okay? So, ganun gagawin natin. Yung conjugate base lang dapat yung matira dito. Anything else na kasama niya should be written sa other side ng equation. How? Okay. So, paano yan? Yung pKa, ilipat mo muna. So, I will move my pKa to the other side of the equation. So, subtract ko lang yan. So, that will be pH minus pKa. That will be equal to the log of the conjugate base over the acid. Okay? Then what's next? Mapansin nyo, yung conjugate base, nasa loob siya ng log. Okay? Nasa loob siya ng log. Gusto natin, ang conjugate base lang yung maiwan. So that means, ilalabas natin siya sa log. Paano uli magpapalabas ng number sa log? Gamit ka ng anti-log. So ano meaning natin doon? Gagawin mo base 10, yung ating number, at itong log function, ito yung gagawin nating exponent. Okay? Gagawin natin yung anti-log. Paano uli yung anti-log? Okay? Gagawin mo base 10. That's yung exponent mo ay kung ano yung given mo doon. pKa yan. So this is 10 raised to log conjugate base over acid. Then what will happen? Yung log pati yung 10, they will counter each other. Magka-cancel sila. So you will be left with 10 raised to pH minus pKa equal to the concentration ng conjugate base over the acid. Excuse me. Since we are looking for the concentration of the conjugate base, yun yung hinahanap natin, conjugate base. Anong gagawin natin dito? Yung denominator natin may acid. Ang kailangan matira, conjugate base. So, we cross-multiply the acid to the other side. No? So, our resulting equation will be conjugate base is equal to acid times 10 raised to negative, pH minus pKa. Okay? So, ito na yung maging resulting equation natin. So, basically, you cross-multiply the acid to the other side. So, conjugate base will be left alone sa isang side. No? So, that's how we manipulate the equation. Paki-note na lang yung mga manipulation sa equation. Especially yung anti-log. Important yan for this chapter. Don't worry, for this chapter lang naman. Anything after that, madali na lang. Back to memorization ulit. Okay. Ngayon lang tayo may solving. I mean, sa chapter 5 may solving din. Pero Excel na lang natin yun.
Sir?
Yes po.
Ano nga po ulit, sir, yung idea? Bakit po kapag ginawa pong base yung 10, tapos naging exponent yung log, bakit po na-cancel po yung 10 raised to log?
Kasi ang log and yung anti-log, no, they are inverse functions. No? So, pag sinabi sa atin na inverse function, magkaiba sila ng domain pati ng range. Yun yung sabi sa algebra. Si logarithm, yung opposite niya, yung inverse niya would be the antilog, no? So parang pag pinagsama mo yung isang function with its inverse, they will cancel each other. Ang pinaka-best basic example dyan would be this one. Suppose x yung ating original function. Tapos, ano yung inverse ng x? 1 over x. Pag pinag-multiply mo yung dalawa, pag pinagsama mo yung dalawang yan, magka-cancel out sila. No? However, for logarithms, exponential yung form nila. So, meaning nun, kapag yung log, ginawa mo siyang exponent ng 10, dun lang siya magka-cancel. So, yun yung counterpart nito in this form. So, parang yun yung other forms nitong equation na to. So, yung x, ito yung original function natin. Let's suppose ito yung log function. Yung 1 over x, this is our inverse function. So, original, then yung inverse, pag pinagsama mo yan, magka-cancel sila para maging 1. Ganon din sa logarithm. Although you do not have to multiply them. So, hindi sila minumultiply. Ang rule sa logarithm ay gagawin mo siyang exponent. Okay? So, kunwari yung 10, ginawa mong exponent yung log, dun sila magka-cancel out. Dun sila magiging 1. Pag yung log naman, ni-raise mo sa 10, dun lang din sila magka-cancel. So, ganun yung batas sa exponential functions. Ayun.
Thank you,
Yun
sir.
lang. Ang daming biglang nag-math tayo. Don't worry, ngayon lang yan. Susunod Excel tayo naman. Chapter 5, mag-Excel tayo. So, basta yun. In short terms, ano na lang, 10 raised to log, cancel out sila. Okay? Basta makita mo yung log, ang gagawin mo dyan, gagawin mo siyang exponent. Ang base mo will be 10. Okay? Pag 10 yan, tas log yun, dun sila magka-cancel out. Yun yung rule sa algebra. Okay? So, going back to the problem, so we now have the final equation na gagamitin natin. So, that will be conjugate base equals acid times 10 raised to pH minus pKa. So, conjugate base, hanapin natin yung millimoles niya na kailangan to have this buffer solution. So, anong acid? So, we have 10 millimoles of acid times 10 raised to the pH that we want. Ang pH na gusto natin ay 5.27 minus the pKa of the acid, which is given 4.752. So, compute lang natin. That will give us the millimole of the base needed. Makikita natin yung millimoles ng base needed. So, that will be 10 times 10 raised to 5.27 minus 4.752. That will be 32.96 millimoles. Medyo maraming medyo maraming conjugate base ang kailangan. So, ang final answer natin would be yung sodium acetate. Ang kailangan natin ay 32.96 millimoles. Ang acetic acid natin, ang kailangan natin ay 10 millimoles. Okay? So, yun yung gagawin natin. So, you will prepare 32.96 millimoles of sodium acetate and then 10 millimoles of acetic acid. Ano lang yan? Mass divided by molar mass. Although, hindi na natin gaganunin dito. Yung basic lang tayo. Ngayon lang natin to gagawin eh. So, ganun lang gagawin yun sa lab. Mag-weigh ka lang ng sodium acetate, mag-weigh ka ng acetic acid. Pag hinalo mo sila, buffer na yon. Yun na yon. Pag haluin mo lang dalawa yung solution na yon. Tapos na. You have a buffer already. So, yan. So, if you want to double check, pwede naman. Burahin ko na tong part na to. Kung gusto mo i-double check, ang pwede natin gawin is we can plug in the info sa Henderson-Hasselbach equation. Kailangan kapag plinag-in natin yung conjugate base pati yung conjugate acid, kailangan pag plinag-in natin ito, pati yung pKa, ang lalabas na pH ay ito. So, let's double check kung yun din yung lalabas. So, pKa is 4.752 plus logarithm ng yung conjugate base sa taas, yung salt, 32.96 millimole. Yung nasa baba ay yung acid, 10 millimole. Solve natin yung pH. So that will be, ayun, 5.27. Okay, so yun. We ended up with the same or with the final pH of the buffer. So ganun lang. Just in case na yung concentration or yung moles ng conjugate acid, conjugate base ang hinahanap, then you have to do the derivations na ginawa natin kanina. Pero kapag yung pH lang hinahanap, just plug them to the Henderson-Hasselbach equation. Okay? So ipa-plug mo lang sila. Pero kapag again, concentration yung hinahanap, magmamanipulate ka pa. Gagamit ka pa ng anti-logarithm. Okay. Next one. Aspirin has a pKa of 3.4. What is the ratio of, excuse me, what is the ratio of the conjugate base to acid in the blood and in the stomach? Pag nakakita kayo ng ganito sa quiz, pag tinanong yung ratio ng A minus to HA, so that is just the fraction. 'Di ba? Maalala nyo yung Henderson-Hasselbach equation. Pag nakita nyo yung sinabi na ratio nito, basically yung fraction lang na ito yung kailangan. So you have to solve for that fraction. Okay? So I will derive, then kayo na bahala mag-plug in ng values. Kung gusto mo hanapin yung ratio ng conjugate base sa acid, so rewrite natin yung equation. Yung pKa, lipat natin sa kabila. So that will be pH minus pKa equal log ng conjugate base over the acid. Then since yung fraction, gusto natin yun yung ma-solve natin, pero nasa loob siya ng log function, then we have to do the anti-log para matanggal yung log. Okay, yung fraction yung gusto natin eh. Tatanggalin natin yung log using anti-log. So that will be 10 raised to pH minus pKa equal 10 raised to log conjugate base over acid. So, magka-cancel yung 10 and log. So, the final equation will be conjugate base over acid is equal to 10 raised to pH minus pKa. So, yan. Yan yung gagamitin nyo pag ang hinahanap ay ratio of A minus to HA. Yung A minus, yun yung conjugate base. Yung A, yung HA, yun yung acid. Okay? So I want you to solve for the ratio of the conjugate acid, the conjugate base and the acid with the following buffer solutions, yung blood pati yung sa stomach. No? So plug in nyo na lang dito, then type in your answers sa chat box natin. So I'll be back. Okay, so let's check yung ratio ng conjugate base sa acid natin. So that means in equation, conjugate base divided by the acid. Okay, for blood and sa stomach, no? So basically, plug in lang natin. So that's 10 raised to the pH, which is 7.4 minus the pKa. So that will be 10,000. Okay, so 10,000 laki, no? So 10,000 ang ratio nila. Then kapag ano naman, kapag acid, kapag sa stomach acid naman, so that's 1.4 minus 3.4, that will be 0.01. So ano yung pwede nating gawin dito sa info na to? Okay. By obtaining the ratio of the conjugate base and the acid, you actually determine kung ano yung amounts ng conjugate base and the acid na kailangan to create the buffer. Ano lang gagawin nyo dito? So yung ratio na makuha nyo, just divide them by 1. Okay? So ibig sabihin nun, you need to have, let's say, for letter B, to create this buffer of solution, kailangan may 0.01 millimoles of the conjugate base ka. And you must have 1 millimole of your acid. So, pwede ganun yung maging interpretation nito. So, that will tell us the ratio of the conjugate base and the acid. So, kung may isang mole ka ng acid, you must have 0.01 millimoles of the conjugate base. So, that means if you have 2 moles of acid, then you must have 0.02 millimoles of the conjugate base. Kung meron kang 10 moles of acids, kailangan may 0.1 mole ka ng conjugate base. So, that will tell you kung ano yung exact proportion ng conjugate base and the acid mo for you to have the buffer solution. Okay? Pero pagdating naman nito sa letter A, yung blood. Okay, so since malayo yung pKa sa pH, so that means malaki talaga yung yung difference ng concentrations ng iyong base, conjugate base sa inyong acid. So if you have 1 millimole of your acid, then you must have 10,000 millimole of your conjugate base. Okay? So kailangan overpowering yung conjugate base nyo so that your pH will increase. No? So ganun lang. Yun yung importance nito. Ano uli yung pwede natin makuha dito? Just divide them by 1. Okay? So divide mo lang yung ratio by 1. So that means if you have 1 mole of the acid, ito yung mole ng conjugate base na kailangan to create the buffer. Okay? Tapos anong gagawin mo para magawa mo yung buffer? Titimbang ka lang ng acid and yung conjugate base. Paghahaluin mo lang sila. Yan, mayroon ka ng buffer. Ganun lang siya kadali gawin. Kaso hindi natin siya magagawa ngayon. Okay. And ito, punta na tayo sa last part. So ano-ano yung mga buffer system sa ating katawan? Okay, so first one, we have the bicarbonate buffer. And the bicarbonate buffer maintains the pH of our blood. No? So, bicarbonate buffer, ito ay medyo acidic. No? Okay, so at acidic environment, the buffer will expel the carbon dioxide, no? So that means mag-exhale tayo. So yun yung nangyayari sa ating katawan. So kapag nagbe-build up na yung carbon dioxide sa ating body, our blood becomes acidic, no? So for the body to to keep the pH high, para hindi siya maging acidic, ang gagawin niya ay i-expel yung carbon dioxide through the alveoli, no? And then palabas ng body, no? By respiration. Pag masyado naman basic yung ating blood, no? So ano pwede natin gawin? Pwede yung carbon dioxide, that is a byproduct of the metabolic process, hahalo siya sa blood, no? Para ma-maintain yung pH. Okay? So kapag sobrang basic ng blood, yung mga CO2 sa katawan natin, hahalo siya sa blood para bumaba yung pH. Pag masyado naman mababa yung pH, ang gagawin ng katawan natin, mag-exhale tayo. Okay? Para yung CO2 sa blood matanggal. So the pH will increase again. Okay? So ganun lang, yun yung balance sa atin. Okay? So that's the bicarbonate buffer. And then the next important buffer system would be the phosphate buffer. Ang phosphate buffer, ito yung mga nasa loob ng cell. Yung ating cell kasi, that is composed of lots of phosphates, no? Maraming phosphate ions doon. Phosphate by phosphate ion, which is because of the production of the adenosine triphosphate, no? Kapag yung ATP na gamit, magre-release yun ng phosphate sa ating cell. Then, gagamitin yon to remanufacture ATP, no? So, yung phosphates na nagagamit and nare-release, no, during those biological processes sa ating katawan, they actually create this buffer system, no? Okay, so they maintain the pH of the blood, no? Okay, so that's why kapag, let's say, cyanide poisoning, ang hina-hijack kasi ng cyanide poisoning ay yung electron transport chain. So it hijacks the production of ATP. So ang nangyayari kapag nawalan ng ATP na yung katawan natin, so maraming factors na pwede maraming processes na pwede mahinto, no? So we know ATP is the energy currency. Without that, the cell cannot function. Other than that, ang isa pang reason is that kapag hindi tayo nagpo-produce ng ATPs, dumadami yung phosphate sa ating cell, which can lead to the disruption ng buffer system. Kapag dumami na masyado yung phosphate, pwedeng masira na yung buffer system. And as a result, any changes sa pH sa ating cell could mean death sa cell natin. So, pwede masira yung cell natin. So, ganun yung two perspectives pag pinag-uusapan yung cyanide poisoning. Pwedeng namatay yung cell because of the low production of ATP, nahinto yung production ng ATP, and the other reason would be the disruption of the buffer system sa cell natin. Kapag nasira yung buffer, patay ang cell. Okay? So, ganun. That's why kapag pinapatay natin yung bacteria, ano ginagawa natin? We use concentrated acids, di ba? Minsan. Kaya gumagamit tayo ng concentrated bases such as bleach. Okay, the purpose is to disrupt the pH, no? So that the cells ng ating bacteria na nandun, mamatay sila. So, pwede rin mangyari sa body natin yun, okay, by just changing the pH. So, that's why we have to have the buffer system to maintain life, no, or to maintain stability. Once the buffer system is destroyed, maraming consequence yun, one of which is death, no, ng cell. Okay, so that's all for water. So, that means quiz na naman next week, no? By the way, next week, Wednesday and Thursday, wala tayong magiging discussion. Why? Kasi, di ba next week yung founding anniversary? I think may schedule sila Wednesday, Thursday na flare session. So, yung flare session, yun ay mga alternative classes, no? Naka-attend na kayo nun siguro. So, I also have alternative class for next week. Thursday yon 3:30 to 4:30. Di-discuss lang ako doon paano mag-adulting. Okay? Kasi based on experience, no? Paano magpalago ng funds, no? Paano maging secured sa buhay, no? Okay. Kunwari, pag namatay ako ngayon, may milyon na ako. Basta mamatay muna ako. So, doon, ikukwento ko yung mga ganong stuff na. So, kung gusto nyo sumama dun sa mga flare session na yun, you can attend, ha? Yun. So, next week, medyo maraming activity yung FEU kasi founding anniversary niya. Okay, so yun lang muna. Ayun, so kita-kita na lang tayo ulit sa susunod. Okay, so that's all for today. So thank you for attending. Pag may question kayo, message lang kayo. Message lang kayo. So yeah. Okay, so bye-bye and